Good evening, ladies and gentlemen. Welcome to the second of many First Animals free evening online lectures from the Oxford University Museum of Natural History. My name is Jack Matthews. I am a research fellow at the museum and very pleased to be introducing Luke tonight. But just before we get going, just to familiarise yourself with the Webinar Jam platform and to say there's a chat function on the right-hand side. So if you click on the chat, you can both send a message to all the wonderful people who are joining us here tonight from all over the world. And we've got friends joining us from Sri Lanka and California, Australia, Newfoundland and Portugal and many other places besides. So do pop something in the chat there. But you can also, if you have any questions at any point for Luke, do pop them in there and we'll have a selection of those asked to Luke at the end. The only other thing to say is we'll be having a few pop-ups popping up there. So there's a link up there at the moment to the next talk by Dr. Imran Rahman and some other links pertaining to the museum. But at that point, I will say that we are very pleased to be joined by Dr. Luke Parry, who is a paleontologist based in Oxford University's Department of Earth Sciences and also a research fellow at St Edmund Hall. And we're very pleased to have him with us tonight to talk about some worms. And I don't want to spoil what is a wonderful title. So at that point, Luke, take it away.
And thank you all very much for coming. I hope you're as excited to hear about worms for the next 40 minutes or so as I am to talk about them. So yeah, let's get going. So I thought I'd start the talk by just introducing what exactly worms are. And this is the definition from Google. So worms are any number of creeping or burrowing invertebrate animals with long, slender, soft bodies and no limbs. So this might immediately make you think of earthworms, but animal diversity contains all manner of different animals, which can roughly be described as being worms. So this includes things which are pretty nasty predators in the ocean. So things like, hang on, I'm just gonna try and draw here. A few more technical hiccups. Okay, so like this fellow here. So this is what we call a chaetognath or an arrow worm. And this here is the business end of this guy, where it has these grasping spines for grasping prey. This thing here is is a type of annelid worm, which I'll be talking a bit more about later, which is another type of marine predator. And then some worms include things which are really, really tiny and microscopic. So things like this nematode here, or these little guys here, which are sorts of animals that live between sand grains that are all around us on sandy beaches, but you probably have never seen them. And then there are animals that are sort of temporary worms that are worms only for part of their life cycle. So this here is an insect larva, and they sort of do their wormy thing for the earliest parts of their life cycle before they become the adult insect here, like here on the right. And then there are more familiar things which are quite beneficial in modern terrestrial environments. So you've probably heard about how earthworms are really important for the cycling of organic matter in soils. And then things which are a bit sinister, like this here, which is a parasitic worm called a blood fluke. So worms encompass all sorts of different animals doing all sorts of different things in modern ecosystems. So when we as biologists, and evolutionary biologists in particular, think about animals, we like to classify them and think about what their close relatives are. So this diagram here, which probably has a load of fairly baffling and unpronounceable names on it, is what we call a phylogeny, or an evolutionary tree. And what this diagram tells us is just how closely related different types of organisms are. So to orient you here, these here are sponges, which unfortunately look a little bit more like this in reality. The organisms here are cnidarians or jellyfish. Things like starfish and sea cucumbers and other closely related things here are called echinoderms. Arthropods, so things like centipedes and insects, are here. So mollusks, so things like snails and clams, are here. And annelids, the segmented worms, which are what I'm going to be talking about for most of this talk, are here. And then we're here, so closely related to echinoderms and other things. But if we look at this diagram and see where worms are, they're all over the place. So the yellow text here is showing all of the different groups of animals that are alive today that we might look at and say, that looks like a worm to me. And then shown here in blue are groups of organisms where some of their representatives are worms for at least some of their life cycle. So for the example of jellyfish, some of their which are here, some of their close relatives have evolved to become wormy parasites. So hang on. So the take-home message of this is that worms are an awful lot of animal biodiversity. And there are lots of worms which are distantly related to each other, all doing their wormy thing independently. So the group that I'm most interested in my own research is this group called Spiralia or Lophotrochozoa, which is the group to which Annelids, the segmented worms, belong. And this includes a lot of different organisms that you might think of as worms. Some of these are microscopic, some of them are predators, and some of them, like Annelids, are segmented. So they have multiple repeated body units. And this is the group that includes earthworms and leeches, but also a big diversity of organisms that we don't see on land, but we do see in the ocean. So when you think of annelids, so these segmented worms, the ones that you might be most familiar with are earthworms, but this group also includes leeches. These are really, really common in terrestrial environments, so environments that are on land. But if we look if we look to the ocean, then there's this huge diversity of form, doing all sorts of different things with all sorts of different lifestyles. So a lot of these animals have easier to remember sort of common names. So sort of based on what their gross appearance is or the sorts of things that they do in the ocean. So this chap here is the green bomber worm. And they're quite interesting in that they have these luminescent green bombs that they keep near to their head that they release to distract predators. There's this thing here, which is called a spaghetti worm, named after its crown of tentacles on its head. There's this here, which is the ice cream cone worm, named for its hard cone-like tube that it secretes to make to live inside. There's this here, which is the Christmas tree worm. So what you're seeing here is just this feeding crown of tentacles that it uses to collect food particles. And then we have this of a marine polychaete called a ragworm. And these are really commonly used in the UK, at least, as fishing bait. So some of you might be familiar with these, which is the same substance that insects and their close relatives use to harden their body. And you can see them all here repeated down the body in the side view of this ragworm here. And these bristles are called chaetae, and this is where they get their name from. So polychaete just means many bristles. So yeah, so these are the organisms that I mostly study, and this is roughly what they look like. So you might be thinking that these animals are entirely soft and squishy, and we know that most of the fossil record is made up of the hard parts of animals, so things like teeth and bones and so forth. So you might not expect annelids to have much of a fossil record at all, and it may not be immediately clear what that fossil record will look like. So before I start talking about the annelid fossil record and exactly what annelid fossils look like, I just want a quick primer on what the history of the planet looks like and sort of the timescales on which we're talking about. So the origin of Earth here is at about 4.5 billion years ago, but it's not until sometime in the last 1 billion years that we get any fossil record of animals at all. So the earliest fossil record of animals is around about 560 million years ago, and most of annelid evolutionary history is contained within the last sort of 540 million years of time. So this is just a quick overview of what the annelid fossil record looks like and the sorts of things that I'm going to talk about for the remainder of the talk. So this timeline, just to the left of where you can see me talking, is the distribution of the different groups of fossils through time. And there are three principal types of fossils that I'm going to talk about today. So the first one I'm going to touch on is these hard fossilized dwelling tubes that many different groups of annelids make. And these are found in the fossil record for roughly the last 250 to 300 million years. And you can see that those are here. So I'm just going to turn that down. Then the second type of fossil that I'm going to talk about is these little jaw elements here, which are made by one particular diverse group of annelids. And then finally, what my research is mostly focused on is these really exceptional and important fossil deposits where we get special circumstances resulting in the fossilization of complete animals in what we call exceptional preservation. Okay, so fossil tubes are made are made by a lot of different groups of annelids, and they've evolved a few different times independently in different not closely related lineages. And probably the most common that we find as fossils are these things called serpulids in a group called Serpulidae. And they make these tubes out of a mineral called calcium carbonate, which is the same mineral that makes up limestone. So these are really, really resilient and resistant to the processes that happen to an organism after they die and start their journey on the way to become a fossil. So we first see these sorts of organisms around about 250 million years ago, and then several tens of millions of years later, they start to form reefs. And they become important in terms of their impact on the biosphere from around 200 million years onwards. And you can see that although the hard parts that we get in the fossil record are only these sort of like cream-colored tubes, the animal in life has this complicated feeding apparatus. So these are include the Christmas tree worms. So they use these complex tentacles to capture food particles from the water column. But another group of annelids that we find quite readily as fossils are things called jawed polychaetes. So there are two main groups of annelids that make jaws. So they make these out of hardening their cuticle to make these resilient structures that we then find as fossils. So they're found in a couple of different groups. So to the left here, it just shows the four different main types of jaws that we find within annelids, within polychaetes. And at the bottom here, you can see that there's apart from the one of the middle sets here, which is C, D, and E, all of these images are from CT scans. So this is a technique where we bombard a specimen with X-rays, which are sort of like having a CT scan in a hospital. And this allows us to reconstruct things in 3D. So these were whole animals that were put in a CT scanner and scanned. But we're only showing the jaw apparatus here to show what the different types of annelid jaws look like. So at the bottom here, we have the jaws from what's called a scale worm. And right here, hang on 1 second. Right here, you can see what the jaws would look like if you were looking directly into the mouth of this annelid worm here. So they have these four jaws in sort of these up and down biting pairs. And the way that annelids use their jaws is by pushing effectively the front part of their digestive tract out of their bodies by squeezing their body wall muscles. So you can see that we have this group here is called glycerids, or the blood worms. So these are quite commonly used as fishing bait in North America, for instance. So some of you might have seen these guys. And one interesting thing about them is that they're quite highly venomous, and their bites can actually be quite painful. So they have these four jaws here, and these are situated right at the tip of this structure here. So the actual body of the worm ends about here. Ooh, hang on, about here, and then all of this structure here is this sort of front portion of the digestive tract that's been squeezed out of the body, and then it uses these jaws to grasp and then pull in food items. So these are pretty bizarre animals, and then sort of not really jaws in the way that you might think about jaws in vertebrates like us, for instance. And then there are these ragworms. So these are another group that are commonly used as fishing bait that have this single pair of jaws. So this is what they would look like front on, and this is what they look like viewed from the top. And again, like the bloodworms or the glycerids, they use these by forcing them out of the front end of their body. And then finally, the most complex jaws that we see in annelids are in a group called eunicids. And they have two sets of jaws. So they have these plates down here. So this is looking at the looking side on at a worm, very much like this fellow here. And so they have these jaws at the bottom and then these jaws at the top. And they form this complex multiple element jaw apparatus, which is used for a range of different things, including grasping prey in groups that are predators. So fortunately, because these, although these jaws are mostly only made out of organic materials, they're quite resistant to the processes that happen after an organism dies. So they're highly resistant to microbial decay, and then they're also resistant to other processes that happen inside sediments as fossils are being formed. So typically, we might if we're looking at a sediment, we might only find a single jaw element like this here, And if we find multiple elements together, then we might be able to reconstruct all of the jaws of a different animal. So that's what we have here, and you can see what these jaws look like when they're being pushed out of the mouth of one of these eunicid worms here. So it's important to note that when we find these as fossils, we typically only find the individual little jaw elements, and then all of the rest of this soft anatomy here is lost to decay. Okay, so now I'm going to show a video of what these guys look like. Okay, so this is a video from the Smithsonian Institution. And these things are extremely voracious ambush predators. So you can see, so this is a fish. And here is one of these worms sitting on the seafloor, sitting and waiting for some prey items. And once they get within range, they strike rapidly and then pull them down into their burrows. So yeah, so these get to very, very large sizes as well. Okay. And we're back to the presentation. There we go. So the typical way that we get these fossils out of sediments is by dissolving large quantities of rocks. So this is a setup here that a colleague of mine called Mats Eriksson uses, and shown in the middle here is what you get after processing several kilograms. And then getting these fossils out involves painstakingly picking through them until you find all of the different elements, like this jaw apparatus that's shown directly above me here. So now I'm going to talk about a fossil that I was fortunate enough to work on from about 400 million years ago, from the early to middle Devonian of Ontario in Canada, from a fossil deposit called the Quattubohagan Formation, which is about 400 million years old, and it's near Hudson. It's in the Hudson Bay Basin in Ontario in Canada, and these are quite unique. So the jaw elements that I showed on the previous slide, so these things here, the largest of these jaws is about 1 millimeter long, and to put that in context, this scale bar that you can see here is also about 1 millimeter. So these things are over 10 times larger than the typical annelid jaws that we find in the fossil record. So these jaws were actually found in 1994 by a team including a colleague of mine called Dave Rudkin, and he showed them to me when I was visiting the museum in 2014. And sort of realizing how big they were, I thought we had something quite special. So this is what the fossils look like. You can see that they're mostly just preserved as these vacant molds, but occasionally, like the fossils that we see, more typically when we dissolve them out of rocks, they also have some black material associated with them, which is the original organic material that formed the jaws when they were alive. So we put these jaws into what's called a micro-CT scanner to see if we could find jaws hidden within the rock that weren't visible on the surface, that might be a bit more pristinely preserved. So I did this at the University of Bristol, and you can see here that we were able to get really, really beautifully preserved individual jaw elements of this extinct predator using this technique, and that allowed us to put together the whole jaw apparatus, and then based on what the soft morphology of of these animals is we were able to reconstruct it as this gigantic ambush predator. And this reconstruction here was done by an undergraduate at the University of Bristol, where I was a PhD student at the time, predating on one of these Devonian fish. And we named this fossil after the bass player Alex Webster from a band called Cannibal Corpse from the United States. So this fossil is really, really unique because unlike all of these other fossil jaws that are known from other fossil deposits, they were extremely large. So when we see living species today, where this first maxilla, which is the jaw element that I showed on the first slide when I introduced this organism, when this gets to over about 12 millimeters in length, then these animals get up to over 1 meter long. So this was really, really a gigantic organism in the Devonian Ocean. And this is completely unique in the fossil record. So although we get annelids that are this size, jawed annelids that are this size today, this is the only instance that we know of when they get this big in the fossil record. And it's important to note that they get this big completely independently of the living ones. So this is an example of convergence, where although they're closely related, they got big independently of each other. And there are another group of annelids that we also find from hard parts that aren't just that are jawed polychaetes, in fact. And this is a group called Machaeridians, the armored annelids. So you can see to the left-hand side of the screen here, this orange material is a load of different individual shells. So there's one of them here. For instance, this is outlining an individual shell. And these are repeated down the body to create an armored organism that in life would have looked something a little bit like this in this model reconstruction. And it was unclear what sort of animal these organisms were until this soft tissue specimen was described in 2008 by my old PhD supervisor and colleagues from Yale University. And this showed that these shell plates that we see on this whole organism on the left here were sitting on a soft body that looks very much like a modern polychaete worm. So this is an example of armored annelids. So here we are in Morocco collecting a few more of these things where they're really, really abundant. And so we were able to pull specimens out of the rocks that look a little bit like this, this reddy orange specimen preserved here. And we took a bit more of a look at a particularly large specimen of these armored polychaete worms in a paper last year. And we found that they had, at the end of their body, which you can see here, so here are all the shells, and then here, sitting at the end of the body, is what we see in these images here. They had these paired jaw elements, rather like what we see in this animal here, which is a modern scale worm. So these armored worms would have been stomping around the seafloor about 480 million years ago, terrorizing other invertebrates and grasping at them with these jaws. Okay. So that sort of brings the portion of the talk that's about the hard parts of annelid worms to a close. And for the remainder of the talk, I'm going to be talking about my primary research interest, which are so-called exceptionally preserved fossils. So these are types of fossil organisms that preserve the components that are typically lost to microbial decay, either after the organism has died and is sitting rotting on the seafloor, or after it's been buried, rotting inside sediment. And in these deposits, we get things which are truly astonishing. So we get preservation of skin, we get the cuticles of these annelids, we get their appendages preserved. So that's either the fleshy appendages that stick out from the sides of the body, which are carrying these hard bristles called chaetae. We also get their sensory and feeding appendages, so like these tentacles that are sitting on the front of this animal here. And we also, in really exceptional circumstances, get preservation of really fine details of their original tissues. So that might be things like fossilized muscles, fossilized digestive tracts and guts, and as I'll show at the end of this talk, even fossilized nervous systems and brains. So these exceptionally preserved fossils are really, really important to us as paleontologists, as people who want to think about what ancient organisms and ancient ecosystems look like, because they tell us completely different things about ancient ecosystems compared to what the conventional fossil record tells us. So this is a picture of a seafloor from about 500 million years ago, showing the sorts of organisms that we typically find as fossils. So these here are trilobites. These here are brachiopods. So typical fossil deposits tell us about the sorts of organisms that produced hard parts, sort of what their species diversity was, how abundant they were, and maybe also what their ecology was. But it's only really when we get these exceptionally preserved fossil deposits that we get insights into what entire animal communities look like. So this is an example from a fossil site called the Burgess Shale. And this is showing an identical scene with all of the organisms with hard parts from the previous slide, but superimposed are all of the diverse groups of organisms that are only known from their soft parts. So these deposits provide really, really unique insights into ancient organisms as well as the ecosystems in which they lived. So the first example of soft-bodied annelids that I'm going to talk about are from a deposit from about 95 million years ago that comes from Lebanon. So laid out down this column showing geological time, what we have here is all of these different stars represent different times in which we have exceptional fossil preservation. And these occur discontinuously, so they're particularly concentrated at about just before 500 million years ago, which is something I'm going to talk about a little bit later. And then one of the youngest fossil deposits is this one here at about 95 million years ago, which is outlined by this white box. So annelids are quite diverse in this deposit from Lebanon, and typically the fossils look like they do here on this image to the left. So what we can see here is these sort of indistinct outlines of the organisms, where they've been quite highly decayed, and in some instances we might only have just the impressions of their hard parts and then a ghostly image of the original anatomy, or just the bristles sitting in their original place in the animal, but with no soft tissue preserved whatsoever. But in one particular species, we actually have all of the muscle tissue from these individual animals preserved. So this is actually 3 specimens of the first fossil species that I ever named back when I was a PhD student. And this white material that you can see here is a mineral called calcium phosphate, which after the organism was dead and buried, as a result of a load of different reactions that were the result of microbial action happening inside the sediment, this mineral has then gone on to replace and finally replicate different tissues, particularly muscle tissue, in these animals. So we can use special light and photographic techniques to really make these features pop out. So shown here on the left is the whole animal, and then these 2 images here show a little bit of a section from the body here. So this image here is just showing what the body looks like if we photograph it under normal light, and then this image here is photographing it under UV light. So what makes these organisms particularly easy to study is the fact that they're preserved as this mineral calcium phosphate. This mineral fluoresces when you light it with UV light, and so it makes the individual morphological details really pop out when we're studying them. So that's one way that we can study these really, really exquisitely preserved annelid fossils. And another way is using what's called scanning electron microscopy. And this is a technique where you can image things which are fractions of a millimeter in size by bombarding them with a type of particle called an electron. So these 4 images here are showing these electron microscope images, and these sort of striated transverse structures that you can see here in high magnification, or running this way in this image in lower magnification, or different sets cross-cutting each here in slightly lower magnification as well, are the imprints of fossilized individual muscle fibers. And the fact that we had this really, really fine scale preservation of these mussels allowed us to reconstruct the fine-scale anatomy of these organisms in unprecedented morphological detail. And what that allowed us to do is to compare it with the mussel anatomy that we see in different groups of worms that are alive today. And that allowed us to identify it as a group of annelids called fireworms, which are these things here down in the bottom left corner. And these are really, really diverse organisms today that live on coral reefs, and they're quite voracious predators. So that one of the things that they do is that they rasp at corals and destroy portions of coral reefs and act as predators on corals. And you can see that they have these white bristles repeated down their body, which gives them quite a nasty sting. So they're quite unpleasant to eat as well. So they're one of the few soft-bodied invertebrates that you might see moving around during the daytime on a coral reef. And then these images here show what the inside anatomy of these guys look like based on CT scanning. So this is a CT scan of a living fireworm, and we were able to compare what we see going on in the mussel anatomy from these images with what we see in the fossils to identify them as a fireworm. So there we go. And they have a few other features preserved as well, which are sort of consistent with this interpretation. So they have this organ here sitting on the tip of the head called a caruncle, which is a type of chemosensory organ that we see here in this living fireworm, also here, here, and here as well. So they have a number of different features that allowed us to identify them as a fireworm. And that's quite important because fireworms don't really have much of a fossil record otherwise. So there are only 2 other species of fireworms known as fossils, and they're from a few 100 million years earlier. And unlike some of their close relatives, they don't produce jaws, so they don't readily enter the fossil record as hard parts. So this was a sort of really unique insight into the deep time or early evolutionary history of this group in the fossil record. And you can see a few other features here preserved in these fossils. So right here, you can see some of the hard chitinous bristles that occur inside the body that they use to support their limbs when they're doing locomotion. So these are really, really exquisitely preserved fossils that have been replicated by this mineral calcium phosphate. Okay. So now I'm going to move on to the earliest evolutionary history of annelids. So going from some of their youngest exceptionally preserved fossils to their oldest exceptionally preserved fossils to a time period known as the Cambrian. And this is around about 500 million years ago. So you can see here on this time scale in millions of years before the present, we have a number of different fossil deposits that preserve soft-bodied animals that represent some of the earliest phases in the evolutionary history of animals. So again, to bring you back to when in evolutionary time this is. So this is just after we first see the fossils of animals turning up in the fossil record at about 560 million years ago. And this is in a time period called the Cambrian, which starts at about 541 million years ago. So this is what the fossil record of things which we think are animals and their close relatives look like. So there'll be a talk in a couple of weeks' time by one of our colleagues at the Oxford Museum talking about these things here, which are called the soft-bodied Ediacaran macrobiota, which are some of the earliest large, complex soft-bodied fossils that we find in the fossil record. But what I'm going to be talking about is fossils that are a little bit younger from what are called Burgess Shale type deposits. So these are these things here, which occur from about 520 million years onwards, which occur just after the oldest trilobites in the fossil record. So probably the most famous of these is a fossil deposit called the Burgess Shale, which was first discovered over 100 years ago by a scientist at the Smithsonian in the United States called Charles Doolittle Walcott. And this occurs in the Canadian Rockies. And so you can see a little bit of what the scenery looks like here. And this is really, really significant in terms of understanding early animals from a paleontological perspective, because it's the first time that one of these exceptionally preserved Cambrian fossil deposits preserving these soft-bodied fossils was found. But since the discovery of the Burgess Shale, there's been discoveries of a number of new similar sites preserving similar organisms in a similar way, such as this deposit called the Chengjiang biota, which was discovered in 1984 and is a little bit older at about 520 million years old. And a number of fossils from this deposit are currently on display in the museum and online on the museum website as part of their first animals exhibit. So I strongly recommend that you check that out if you get a chance. So I've been fortunate enough to work on fossils from the Burgess Shale, both based on museum collections and going out into the field. So these are a few photos of a field season that we did in August 2018 at a new site which is closely related to the Burgess Shale in the Canadian Rockies. So you can see just above me, we have our campsite, which was our home for 4 weeks. And then you can see sort of what life was like in our field site. So this is the inside of our food tent up on the top left-hand slide. This is a picture of me working in the field using a jackhammer for the first time. And I also got my first ride in a helicopter, which was quite exciting. And then sporadically scattered around are a couple of pictures of some interesting animals freshly split out of the rocks and collected in the field. So all of these are arthropods. So up on the right, we have an example of a trilobite. And then to my left are some arthropods that are only soft-bodied. And you can see their preservation of things like their grasping appendages and their walking legs as well. So really beautiful preservation. And it was great to see these fossils in the field as well. So what I'm really interested in thinking about from these really fantastically preserved soft-bodied fossils is exactly what they tell us about evolutionary history. So I'm thinking about how we can use these different fossils, these ancient animals, to tell us about how the different groups of living animals are related to each other and where their body plans came from. So the Burgess Shale contains a number of really, really weird extinct body plans, which sort of were a bit perplexing when they were first described. So this fossil here is something called Hallucigenia, which has these sort of tentacly limbs and as well as this row of spikes. It was originally reconstructed as walking on stilts, but more modern reconstructions turn it upside down and have it walking on these fleshy legs here. There's weird things like this thing called Opabinia here, which has 5 eyes situated on the top of its head, and this weird grasping claw at the end of a prehensile proboscis. And then these really iconic things called Machaeridians, which are these apex predators from 500 million years ago, which have these sort of scary-looking grasping appendages situated at the front end of their body, which you can see in the fossil specimens and this reconstruction here. And these have had a sort of interesting history in terms of how we think about these ancient animals. And in this book called Wonderful Life by Stephen Jay Gould, he emphasized that these things were very, very different to the sorts of animals that we find in the modern ocean. And he considered them to be sort of extinct body plans, as if this was a sort of experimental phase in the early evolution of animals, coining the term weird wonders for these experimental lost body plans. The way that we think about these now has changed a little bit, and we think about them more in terms of where they might sit in these things called phylogenies or evolutionary trees. And in modern considerations of these weird fossils, we think about how they're related to the living body plans. So they're accommodated as what we call stem groups. So stem groups just describe organisms that sit outside the living diversity of animals that we have today. So an example here from animals which are perhaps more familiar would be the relationships between dinosaurs and birds. So this is the crown group here. So that describes, this here describes the last common ancestor and all its descendants of an emu and a pigeon. So this is the crown group of birds. And then the stem group is all of the different extinct organisms that we think are more closely related to birds than they are to any other living animals. So dinosaurs are more closely related to birds in this example than they are to crocodiles. So they would be the stem group of birds. And thinking about what this means for thinking about these weird animals from the Cambrian, things which were sort of maybe a bit more baffling when they were first described, can now be thought of as belonging to the stem groups of living groups of animals. So this is an evolutionary tree of arthropods. So that's animals like centipedes and insects and crustaceans. And these giant apex predators are now thought as lying on the stem group of the living arthropods. So this that I've outlined here is a Machaeridia, so this apex predator, and then this is the living diversity of arthropods. And this has really changed how we think about these fossils and what they tell us about evolutionary history. So as you know, my research is mostly on fossilized annelids, so fossilized segmented worms. So this is an example of what these fossils look like from the Burgess Shale. So this is a fairly large example of a polychaete worm called Canadia spinosa. And you can see that they're absolutely exquisitely preserved. So we can see that the body is preserved here, and you can see a number of appendages and bristles sticking out from the side of the body. And these are really, really uniquely preserved in terms of the level of morphological detail that they give us compared with other annelid fossils. So this is a couple of examples of different worms that we find from the Burgess Shale and similar deposits. So at the left here, we have an animal called Kootenai scolex, which was described in 2018. In the middle is an animal called Burgess-Shale, named after the Burgess Shale. And then on the right is an animal called Canadia, which I showed on the last slide. This sort of very, very bristly, flamboyant-looking worm from about 505 million years ago. So when we're thinking about these worms, I'm typically thinking about what they tell us about the earliest phases of the evolution of the annelid body plan, and what they tell us about perhaps what the last common ancestor of annelids might have looked like. So when we look at the evolutionary tree of annelids, things that we think of as being quite primitive, that are sort of forming the deepest branches in this evolutionary tree, contain a number of different strange groups of annelids that live in a number of different ways, which includes living in hard dwelling tubes, like this thing called Noweniid, that's sitting at the left-hand side of this slide here. Things in the middle, which are living in burrows or in lined burrows like tubes. And then other things which are actively crawling around on the seafloor. And if we look at the annelids that we get from the Cambrian, they look a lot more like the sorts of animals that are actively crawling around on the seafloor. And we think that based on this, that probably the last common ancestor and the earliest representatives of this large and diverse group of organisms was probably crawling around on the seafloor and not living in a burrow like some of the primitive representatives do today. And this is sort of borne out in thinking about where they go in the annelid tree of life. So this is just showing a tree from one of my own papers here, showing where in the annelid tree of life these things go. So they're sitting down here in the stem group of annelids, whereas the bulk of the living diversity of annelids is sitting up here. So these fossils then can tell us something really, really interesting about the earliest phases in annelid evolution and perhaps what the annelid ancestor looked like. Okay, so moving on. So before I talk a bit about some of the soft tissues that we get preserved in these animals, I just want to talk a bit about how we actually visualize them using different microscope photographic techniques. So this is an example of the fossil that I showed earlier that I was holding in my hand. So this is a fossil that was collected over 100 years ago from one of the original expeditions to the Burgess Shale of an animal called Canadia. And on the left-hand side, you can see what happens if you photograph it just under normal light that's directed straight at the specimen. And you can see that there's not really much contrast. It's hard to see some of the details. The image at the middle, you can see what happens if we dunk the specimen in water, and that really bumps up the contrast as just the bits of the soft tissue and the soft anatomy of the animal reflect the light back. And then on the right-hand side, this is an example of a technique called cross-polarized light, where we're taking only a fraction of the light that hits the specimen, and that makes all of this reflective stuff go black, and that makes it a lot easier for us to observe some of the features that we might be interested in. So one of the fortunate things about the Burgess Shale is that we have these organisms preserved in different orientations inside the rocks. So the specimen that you can see at the top here is an organism that's lying flat on a bedding plane. So what you're seeing is the top surface of the animal. And then immediately beneath that, we're looking at the side view of the animal. And this is really, really helpful because although these animals are paper thin today because they are in rocks that have been compacted down over millions of years to form the rocks that they're in today, it allows us to look at these animals and reconstruct them in 3 dimensions. So this is just an example here of what the head of this thing looks like in different orientations. So again, on the left-hand side here, we have just a top view, which is showing this pair of tentacles and then these bristly appendages repeated down the body. And then the side view is showing what the head looks like side on. So it shows how these bristles are arranged on the body and exactly how all the appendages connect up as well, which is something that is really, really important for understanding what these animals are and are useful characters for evolutionary biologists to study. So that allows us to reconstruct this animal in 3 dimensions, and now that we have a lot more specimens of these things to go on, we're able to update the reconstruction of exactly what this animal looked like. So this is a reconstruction at the top that was done in 1988 for Stephen Jay Gould's book Wonderful Life by Marianne Collins. And then by the same artist, we have this updated reconstruction at the bottom, which shows a sort of similar story, but a slightly different morphology for some of the different bits. So that we now know a lot more about where these tentacles sit on the body. We know that there's one tentacle sitting in the middle of the head that we didn't know about before. So we know a number of different new things about exactly what this animal looks like. And so we're able to see what these things look like in 3 dimensions. And this is an example of a 3-dimensional reconstruction done by a 3D artist called Lars Fields, where we can reconstruct how this animal moved on the seafloor and what its morphology looked like, including bits of its soft anatomy that are usually covered by these bristles, like the gills that you saw in the body there. So I'm just going to go back to the slides. Okay, there we go. So a really special and unique thing about these Burgess Shale fossils is that some of them preserve the original carbon that made up parts of their bodies, and we can visualize that in different ways. So these different images here show a little gill that sits on the side of the animal, and we can use special microscopes called SEMs, or scanning electron microscopes, to visualize the original carbon. So in one of the images here, it looks very black, because it has a low density relative to the rock. And on the image just to the immediate right-hand side here, you can see it's occurring bright red, where we're mapping only the distribution of carbon in the specimen. And when we were looking at these specimens originally, as part of this project to redescribe the morphology of this animal, we noticed that these very, very dark black features were concentrated in different regions of the body. So this is an image here of one of the fossil specimens, and you can see that there's this concentration in the head region here, sort of forming this ring, and also forming on the insides of the tentacles here. And this is sort of very intriguing because it looks a lot like what we see in the nervous system of living animals. So the image here on the right-hand side is from a paper describing the nervous system of one particular polychaete called Neanthes. And what's being shown in red here is where they've used this special fluorescent dye to highlight the shape of the nervous system. And you can see that there's and this is the head region here. And you can see that there's this very close similarity between what we see in Canadia and this sort of loop-like region that we see in the nervous system of extant annelids. And this is the region that we have the brain and also the mouth region as well. So we decided to investigate this in a little bit more detail. So this is an example of elemental mapping, where we've used electron microscopes to map out the distribution of carbon. And you can see that there's a very close correspondence between what we see in these ancient fossils and what we see in living organisms. So you can see that there's this loop that forms here, situated here, which we can see here in this animal. There are these branching structures coming off that go into the limbs and the other appendages, like we can see here in the living animal. And we can also see these very, very bright red carbon patches here that are going into the tentacles, which are very big in Canadia, this fossil, but a lot smaller in this living example. And we were able to show that these different features preserve consistently across different specimens. And we were able to use this to reconstruct what the nervous system of this ancient animal looked like. And we found preservation of the nervous system not only towards the head region, but also in the rest of the body here. So these images shown here show the trunk of this animal, and sort of lying along here is what we call the ventral nerve cord. So this is the part of the nervous system that sends nerve impulses to the rest of the body to control things like locomotion. And you can see that sort of along the body of this living worm here. So this is a really, really unique discovery. Nobody had ever seen this in a fossil polychaete worm before. But people had described fossil brains from other animals. So this is an example of a fossil brain from an arthropod called Fushenhuia, which was described by Xiaomar and colleagues in 2012. And they were able to sort of see really, really important details of the nervous system and the brain in this animal. And we now have fossilized examples of nervous systems from a number of different animals from across from a number of different sites within the Cambrian. And it seems like this is something which is really, really unique to these special Burgess Shale type deposits that we get around 500 million years ago. So we were able to use that then to think a bit about what the evolutionary history of the nervous system was within annelids. So this is just like the very, very rough sketches of what the nervous systems of some of these more primitive groups look like. So we have examples of animals that have very, very simple nervous systems. So this is the nervous system of, I think, the Sipunculum, where they don't have these complex repeated units like we see in something like an amphinomid or a fireworm. And so we have sort of other varying levels of complexity as well. So we see fairly simple nervous systems in also these things called the paleoannelids or the oeniids and the megalonids. Whereas in Canadia spinosa, we see quite a complex arrangement of the nervous system. And on this basis, we reconstructed the last common ancestor of annelids as having a complex nervous system, which has become simplified in some of the organisms which have now much less active lifestyles. So doing things like burrowing or living in tubes. So that sort of wraps up everything that I wanted to talk about. So going from fossilized jaws to fossilized brains. And I'd just like to take some time to thank all of the people that I've worked with over the past couple of years, and all of the institutions that have hosted me, either as a PhD student or as a postdoctoral scientist, and everybody who's helped out with technical assistance and with artwork and reconstructions as well. So again, just to summarize, although annelids are these completely soft and squishy worms that you may not think of as having a really, really good potential to preserve as fossils, we have a number of different instances in which they do preserve really readily. So either as hard fossilized dwelling tubes, as these fossilized jaws, or in really, really unique circumstances as whole animals with their soft parts preserved. So thank you very much.
Thank you very much to Dr. Luke Parry for taking us through over 500 million years of the wonderful world of worms. And just before we move on to questions, and we have lots of questions from you at home today from all over the world, just to say there should be a little box on your right-hand side now popping up, allowing you to sign up for the next talk, which is 2 weeks today, given by one of our fellow researchers, Dr. Imran Rahman, who is Deputy Head of Research and a Research Fellow at the Oxford University Museum of History. And if you have been intrigued by some of the three-dimensional modelling techniques that Luke has talked about tonight, well, Imran's going to give you more of an in-depth view of some of the wonderful techniques that are being used to look inside and discover more about fossils using three-dimensional techniques. So do click on that on the right-hand side. And with that, we will get on to some questions. The first one is not actually a question, but it's worth just mentioning that Jessica noted that her 6-year-old daughter just told her that polychaetes are adorable. So, Luke, you've already had some impact there. Guillermo asks, you've mentioned that the black material associated with the fossils is part of the original organic material. Is it possible to get any information about the proteins of the jaw or something similar?
So there are instances where we have organic preservation in fossils where you can get the original preserved macromolecules. So those are the big structural molecules that make up different tissues. And these different compounds have different preservation potential when they enter sediments. So certain different molecules will decay very, very quickly and disappear either through microbial decay or through processes that we call maturation, which is what happens to sediments as they become rocks. So some of them don't last very, very long at all. So we're thinking sort of the order of thousands of years for things like DNA. And then other compounds like lipids survive for a very, very long time in rocks. So some early evidence of different groups of organisms from before we get large complex fossils are actually from a preservation of things called lipid biomarkers in the sedimentary record. So in the instance of most of these fossils, for most of them, the organic material that's made the jaws has been very, very heavily modified by fossilization. So you probably wouldn't be able to get much information about the about the original proteins. And for the examples from the Burgess Shale, they've been to what's called green schist metamorphic facies, which means that the rocks have been buried very, very deep and heated up and squeezed, which modifies these organic materials as well. So you can get information on the original molecules from some fossils, and people have done that for things like fossil color, so you can detect pigments like melanin from things like feathered dinosaurs. But for most of these fossils, because of their age and the sorts of environments that they were buried and fossilized in, you probably wouldn't be able to do much with the original organic material. So we're not going to see like a Jurassic Park of early worms, unfortunately.
We have another question from Hiroki, who asks, were all of these annelid fossils preserved under anoxic conditions? And what were the depositional facies, i.e. environments, at which the bodies were preserved?
So there's a couple of different places that you get these soft-bodied fossils preserving, and they mostly are associated with anoxia, because that's important for suppressing the bacterial organisms that decay these soft tissues. So they don't all occur in the same depositional environment, and that can actually be quite varied. So I don't know if I can turn the whiteboard back on. I can. Okay. So a couple of these different examples that we can see on this end slide here. So these three were preserved in iron carbonate nodules in quite shallow water from a fossil deposit called Maison Creek. And the setting where the Burgess Shale type deposits occur in much deeper, they're occurring in these sort of event deposits where large volumes of sediment are being dumped by events like storms that are burying these animals alive and preserving them. So although we need similar conditions, like the sediment needs to have a very fine grain size, and we need anoxia, we actually get them in different depositional environments. So things like the muscly worm that I showed sort of earlier on in the talk, that was preserved in a type of rock called a platen calc, which are these fine-grained carbonate rocks. And it's in those sorts of deposits that we particularly get preservation of things as calcium phosphate. So they occur in all sorts of different places, but anoxia is probably one of the unifying factors that results in soft-bodied preservation.
Now we have a question from Denver that's about some rocks that are just a bit slightly older. They're about the end of the Ediacaran 540, 550 million years ago. Denver asks, do you agree with the annelid affinity of the cloudinamorphs rather than them being cnidarian?
Yeah, so that's actually a really, really interesting question. So these for people who aren't familiar with these fossils, these are sort of these some of the earliest hard parts of things that might be animals are these things called cloudinamorphs, which look superficially a little bit like serpulid worms. So they look a little bit like these fellows over here. So they're also mineralized, so they're probably originally made of calcium carbonate, although some of them might have been less heavily mineralized than others. And there was a recent paper earlier this year, I think, that describes some soft tissues from these ancient tube fossils that are about sort of like 545 million years old. So about 20 to 30 million years older than some of the annelid fossils that I showed. And what they showed in that paper was the preservation of what looks like a tubular gut running down these tubes. And that's sort of the authors of that paper said that might be indicative that these things are organisms that are bilaterally symmetrical. So that is everything from us to all of these different wormy groups that I was talking about earlier. Whereas they've been more typically considered to be a cnidarian, which are jellyfish and anemones and their close relatives. And this paper was really interesting because we'd never seen soft tissue from these organisms before, and they reconstructed it as maybe being something like a tube-dwelling annelid. Typically, there are a few problems with that in terms of the morphology of the tubes. So they don't really closely resemble the tubes that we see in any living groups of annelids. So they don't really closely resemble serpulids. And they also don't leave a continuous fossil record after, I think, about sometime in the early Cambrian. So if they are annelids, they're probably a completely independent experiment in creating this tube-dwelling body, this tube-dwelling lifestyle. And it's difficult to tie them to any living group that makes tubes. So I would say that the jury is still out on that, and they certainly don't preserve any features that really would nail them as annelids, like having parapodia or having bristles. or any of the other sort of diagnostic annelid features. So yeah, that would be my answer to that.
And we have a question that's a very similar question, both from Kelsey and from Thomas. Do you think the origin of these annelids lies before the Cambrian? And if so, what would you want to see in the Vendobionta, that's the group of organisms we're seeing in the Ediacaran just before the Cambrian, to identify them as annelids? So do you think there were annelids before the Cambrian? And what would you be looking out for in the weird beasties we see in that time period to identify them?
So that's also a really great question as well. So annelids' close relatives include things like mollusks and brachiopods, and all of these organisms show up unequivocally at around about sometime between 520 and 540 million years. And one of the things that unites all of these different groups of organisms is having these hard chitinous bristles. So some mollusks have them. So things called chitins have these hard chitinous bristles. Polychaete worms have them, and then brachiopods have them as well. So there's a fossil from the late Ediacaran called Kimberella, which has been interpreted as a stem group mollusk. And if Kimberella is a stem group mollusk, so if it is an early mollusk, that suggests that the split between annelids and mollusks has already happened by the time we get Kimberella as a fossil. So if people accept that interpretation as Kimberella as a mollusk, it means that annelids must have been present, at least in very primitive or early forms. But Kimberella is obviously extremely controversial in terms of what it actually represents. So certainly, if they're not occurring in I would probably not be surprised if there was something very annelid-like in the latest Ediacaran. And there's a trace fossil that was described later, sometime in 2019, that looks like it could have been made by a segmented organism, which could potentially have been made by an annelid. But for me to really look at a fossil and think like that's definitely an Ediacaran annelid, I would want to see this sort of like serially repeated segmented body with these hard chitinous bristles, and maybe also some of the other features that unite living annelids. So one thing that primitively annelids had was this single pair of tentacles that sat on the head. So I'd quite like to see those as well, to distinguish it from some of these other bristly groups that might have been around in the earliest Cambrian and latest Ediacaran.
Excellent. We'll take one or two more questions before we wrap up. Just to say, if you're interested in the topics that Luke was raising about this, the divergence of different animal forms during the Ediacaran and the early Cambrian, do go on to the Oxford University Museum and Natural History website. There's a dedicated webpage for first animals. We showed the link before, and you can find more about this very interesting piece of Earth history there. But we'll move on to the next question by Beth Richards, who says, can you talk a bit more about those bonkers 4-part jaws? I'm trying to visualize how they work. Is each jaw piece one corner of a square that sort of expands when the mouth opens? Open brackets, I can't even deal with the giant worm jaws.
Yeah, so what these jaws are like so the cuticle, the skin that lies on the outside of the body is like continues on to the front bit of the digestive tract. And individual bits of that digestive tract have been hardened by different materials. So things that we call sclerotized proteins is what they're typically made of. So these hard materials, that unlike our jaws, they're not sort of like connected together. So they're able to move more or less independently of each other. So when the pressure of the muscles contracting forces them out, they're all able to expand outwards more or less independently of each other. And some of them have like little articulations that joint some of them together. So there's like one pair of jaws that articulates together. So those come out like this when this part of their digestive tract is forced out of their body. But yeah, they're a bit difficult to imagine how they might work unless you sort of take a look at them moving in the living animals. Yeah, they're definitely like a little bit baffling.
And Kayla asks, a lot of these worms look fuzzy or hairy. Are they fuzzy in a way that we understand today, or are they more like venomous spines or barbs? How much is known about the function of these types of structures? And then they add, thanks for a great presentation.
Okay, yes, that's a really good question as well. So in the fireworms, these hard spines effectively mostly for protection. So they're quite unpleasant to handle. If you were to pick one up, you probably have a bit of a bad time. So it's difficult to infer exactly what the function was in the extinct ones, but certainly they perform a load of different functions in different living groups of annelids. So some of them are actually used as anchoring structures. So some of them have like little hooked bristles that stick out from the side of their body that allow them to grip inside tubes. And some of them use them for walking around. So they use them for gripping a substrate when they're stomping around on the seafloor. And others of them also do use them for protection. And in these very, very ancient ones, they look a lot more similar to the groups that use them for protection today than they do like the other functions. So although we can't make observations on these animals alive, we can compare them to sort of similar organisms that we see in the modern ocean. And it looks like they're probably used for protection in these really, really ancient examples. So yeah, that's a great question.
Excellent. I think at that point we will wrap up. Thanks once again to Luke for a wonderful, wonderful talk. And thank you to everyone for joining us today. If you have any comments, any feedback, do please pop it on the chat and we will take that on board and respond to that. Thank you very much. And as I say, do sign up to Dr. Imran Rahman's talk, which is 2 weeks today and should be a very, very interesting look at the wonderful world of the amazing techniques used to make 3D models of fossils and how that reveals a new window on paleontological research. So thank you once again for joining us. We will leave the system running for a little bit longer so people can chat if they want to. But thanks once again for joining us, and hopefully we will see you again in 2 weeks' time.
Brilliant. Thanks, everyone, and thanks for having me.